Hello, today we're going to do the B3 biology paper, and it's the 2014 one, the one from last year. So we're going to be able to go through the B3 paper, learning some exam tricks along the way. So let's get started. So for question 1, we have it talks about haemophilia. So haemophilia is a recessive sex disorder. The family's pedigree shows the inheritance of haemophilia. So it gives you affected male, carrier female, unaffected male, and unaffected female. So the first thing it says, state the sex chromosomes of person B. So we have to look at person B, and then you have to put the right answer. So we know it's going to have to be XX, and this is because person B is an unaffected female, because it is actually carried, it has to be carried on the X chromosome, and because she's unaffected means an XX. If it was a male, it would be XY, and if they were, if they were affected, they'd have haemophilia. Haemophilia is recessive, so it'll be X. It will be X, and then a small h here, like this, on both of them. So I just wrote the answer right now, and it asks, explain why the male offspring from A and B do not have haemophilia. So we need to look at A and we need to look at B, and we need to know why the offspring, the children, don't have haemophilia. So because they did not inherit haemophilia, the allele located on the X chromosome. Now, because you have one affected and one not affected, it's a recessive disorder, so you only need both of them to get it. But this, it shows you here because there are carriers, but they're not affected. Now, this is because they don't have it full, and that's why they do not have haemophilia. So here, it's a common question, and it asks you to do a Punnett square, so to calculate the probability of individuals C and D could have a child with haemophilia. So we have to look at C and D, which is right here, so I just got it from the other page, and we're going to have to write it in. So we have, the first thing we have is C, and it is an unaffected male. So a male is X and Y. And we know they are not affected, so they're going to have a capital H, and Y, nothing's carried on the Y chromosome. And then you have the female, and it's a carrier female. So they do have one, X, which they have one, so a small, and then they have a normal one too. So we just do this, and this will be X, H, so we have two X's, one small, one big. Sorry, there's no space here. And that gives you the answer. So if you have to write something, it shows you, so if they could have haemophilia, so which one does have it. So let me get a different colour. Let me get this colour. And you have to see, so it has to be 25% chance. Can you see that? So here, it's the next part of the question. It says haemophilia can be treated using a blood clots factor produced by a fermenter. The conditions inside a fermenter has to be carefully controlled. Explain one named condition must be controlled in a fermenter. So there's loads of different things you can talk about, and you can get the marks in lots of different ways. So the ones I put, the one I put, is because it's 2 marks, so you explain it, and then you say why. So the temperature has to be controlled for optimum growth to prevent enzymes denature. This is the temperature. So naming any of these factors you have to give, and then you say why, and then it'll give you 2 marks. So explaining what it is and why you use it. So for this question, you need to learn everything about monoclonal antibodies, just in case this comes up, and it frequently does come up. So the diagram shows the stages in the production of monoclonal antibodies. So it shows you the antigens injected inside the mouse, and then it makes B lymphocytes, the myelin cells, which is actually a tumor cell, and then it shows you the monoclonal antibody. So it says complete the sentence by using words from the box. So you have memory lymphocytes, exponential, which we think might not come up in this topic, hybridoma, and immune, antiseptic, and myelin. So injecting antigens into the mouse produces an immune response. So I just wrote this down, immune response. Response resulting in production of antibodies and memory lymphocyte. So they're very nice for giving you the actual possible answers, which is pretty good because sometimes they don't. So the next question says, complete the sentence by putting a cross in the box next to your answer. The cells produced by B lymphocytes and the myelin cells combined are antibodies, hybridomas, memory lymphocytes, or platelets. And the answer is hybridoma. And now this is because myelin cells and B lymphocytes make a hybridoma when you fuse them together. So just learning the stages will really help in this question. So, describe 2 ways which monoclonal antibodies are used in medical diagnosis. So there's more than 2 ways, but we only need to mention 2 to get the 2 marks. To get the 2 marks, you would need to say they're used for pregnancy tests. Now we know this if sometimes it comes up as a 4-mark question saying how does pregnancy test work. So I suggest you revise that. And monoclonal antibodies are also used to locate a position of a blood clots. Now all these 2 ways are actually mentioned in the GCSE book of which you should check out. You need to read it so you can know, just in case they come up as longer styled questions. So for this question, we're going to have to look at the graph. So the graph shows antibody concentration in a mouse after a second injection of the same antigen. So when you know, if you have some certain level of understanding of this, you can see in the first injection, the level of antibody concentration was slower. You can see how much slower it was compared to the second one. This is because of memory lymphocytes. So if we look here, it just says to compare the antibody response after the first injection with the antibody response after the second injection. So it's only 2 marks, so the only thing you have to say is the first response, there's a delay in production of antibodies. However, in the second response, there's an increase of antibodies. So you don't need to have any scientific knowledge in this question here, just because it's just comparing. But if you want to know, it's because of the memory lymphocytes. And because the body actually recognizes it, makes the antibody quicker because it remembers. And that's the answer. So complete the sentence by putting a cross in the box next to your answer. Injecting patients with antigen forms the basis of vaccination. This was first developed by, so they give you 4 different names, which are all valid in the scientific community. So here you have Diane Fossey, which actually she studied behavior in gorillas, so it can't be her. Edward Jenner is the correct answer because he did the vaccination with the smallpox and the cowpox, and if you learn it, you would know. And Louis Pasteur was pasteurization, so it can't be that either. But knowing, it tells you about Edward Jenner and what he did to know about antibodies and vaccination. So for this question is question 3, so we're swiftly moving on. The diagram shows the number of hours of darkness and light needed for flowering in 3 different plant groups. So they give you a short day plant, a long day plant, and a neutral day plant. So here they give you lots of relevant information here. So there's light, darkness, and light or dark. And the question states, state the number of hours of darkness needed for long day plant to flower. So you just have to interpret the information, and you can get a mark between saying anything between 8 to 10. So I'm just going to put 8. And this is from a long day plant, so you look at long, and flowering, light, and then it just gives you the information. So here you have a maths question, so make sure maths is up to point. So make sure you revise maths as well as science. So here it says a garden has 500 short day plants. 85% of these plants flowered in March. Calculate the number of plants flowering in March. So the only thing you have to do is 500. This is the easiest way I do it. So 85% is equivalent to 0.85. And that gives you 425, I believe, which is the correct answer. So here, suggest a benefit of a plant being day neutral. So just common sense. The plant can flower all year round. And the next one is saying plants flower to respond to the number of hours in darkness and light is example of. So you have to know it's not going to be B or A, and it's either going to be D or C. And phototropism is actually to do with the plant bending, depending on light. So number of hours in darkness is going to be C. So the leaves in some species our bamboo plants contain the poison cyanide? Suggest how the bamboo plants benefit from having poisonous leaves. It's only 1 mark, so we only need to state. The answer is they were less likely to be eaten by animals and herbivores. That's it. That's all you have to say. 1 sentence, line, and that will give you the mark. The next part of the question says, the photograph shows a greater bamboo lemur. The great bamboo lemur is adapted to tolerate the cyanide. This means it can eat the bamboo leaves containing cyanide. Explain how the co-evolution of bamboo plants and the greater bamboo lemur could have evolved. So, it's 2 marks, so we need to give a state and explain it. The bamboo plants produce cyanide, survive to reproduce. The mutation in the greater bamboo lemur allowed them to tolerate cyanide. So, 2 ethologists, they studied the call types of communication between the greater bamboo lemurs, and then it gives you some information, and it's another multiple choice question. So the answer would be aggression. I didn't even read out the question, sorry. Complete the sentence by putting a cross next to the box next to the answer. So the behavior associated with the most types is aggression, and you would have to see that from the information and know it. Explain why the benefit of communicating using sound rather than facial expressions. So I would say either because of long distances or because you can't see in the dark, as in some animals, but also you don't need to have any visual contact. It allows communication with more animals. Question 4 talks about human evolution and behavior, and it's another tick box question. It's only worth 1 mark, so don't spend too long on it. It says the species which have thought to inhabit the Earth for the longest period of time is, here's a timeline, it tells us some information, tells you which one. Now when you look at it, which one's for the longest period of time? It's this one. So you're just going to put this answer. So estimate the number of years they inhabited the Earth, and then you just look at where it is, and then you have to, you can have a range between 0.7 and 0.9 million years. So you just have to interpret the information for 1 mark. For the next question, it says scientists in Africa discovered a part of fossilized skull. The skull was identified belonging to the species Homo erectus. Describe methods scientists may have used to identify the skull belonging to the species. So they would look at the shape of the fossil. They could also see the age of the fossil in the location of the rock layer. That's the 2nd mark. So the 1st mark would see the shape of the fossil. And then you can say the structure of stone tools, because as they developed and their brains got better, or bigger, they had better stone tools. Homo sapiens evolved in Africa, then migrated to other areas of the world. Mitochondrial DNA analysis can be used to map human migration. Compare the benefits using mitochondrial DNA rather than nuclear DNA to map human migration. To get the marks because you need to explain 3 things for 3 marks. So mitochondrial DNA is more abundant than nuclear DNA, so there is higher mutation rates, meaning they're less likely to degrade, but also scientists can interpret the information from mutations in mitochondrial DNA, especially that mitochondrial DNA is only from mothers, so it's not mixed with the DNA from the father, so it's easier to interpret. And the next question, one of the group Homo sapiens migrated to an environment where there was a large waterfall. At first, the noise of the waterfall kept them awake at night, but then they got used to the noise over time. Explain this behavioral response. It's habitation, I can't say it, sorry, is a land response. This means they can stop responding to the harmless signal. This is done through horses as well, police horses. I need to get this for the 2 marks. So question 5 is about hormonal control. The diagram shows the body's response to dehydration. So dehydration, you have the brain, you have the hormone response. So the brain, there's thirst, hormonal response, and there's a kidney. And it says use the diagram to help explain the body's hormonal response to dehydration. So here you're going to have to learn about osmoregulation, which is part of the spec, and then you need to know about the hormones such as ADH and how it all works. So this is what you would write. So to get the 3 marks, you would need to say 4 marks, sorry. You need to say these following things. So dehydration is detected by the pituitary gland, and that's like the back of your brain, in the brain. And this causes more ADH to be released, which acts on the nephron. The kidney tubules become more permeable to water, so more water is reabsorbed into the body. So you're thirsty, so more water is reabsorbed back into the body, so you have enough water. Now, this is the key part. You need to say that to get the rest of the marks. So I'm just going to explain the kidney and osmoregulation and negative feedback. So the amount of water reabsorbed in the kidney and nephrons is controlled by antidiuretic hormone. And it's shown here, ADH. And the brain monitors the water content of the blood and instructs the pituitary gland to actually release ADH into the blood according to how much is needed. It releases, of course, all the time, either when there's water too high or when the water is too low. The whole process of water content regulation is controlled by the mechanism called negative feedback. Now, this is where the changes in environment trigger responses and counters the changes. So, this all means that the internal environment needs to stay around normal level where the cells work best. So, now I'm just going to explain about if you have drank too much water or not enough. So, if the water content is too high or too low, negative feedback brings it back to normal, this mechanism. So let's say that you're hydrated and you haven't drank any water. This means the water content is too low. This means the brain actually detects the water loss and the pituitary gland releases more ADH. This actually results in ADH makes the kidney reabsorb more water, which results to concentrated urine. Now let's say the reverse happens and the water content is too high. You've got a gain of water. This means the brain detects the water gain and the pituitary gland releases less antidiuretic hormone. This results in the lack of ADH means the kidney reabsorbs less water, which means that you have less concentrated urine. On to the next question. So, the menstrual cycle is also controlled by hormones, including progesterone. Complete the sentence by putting a cross into the box next to your answer. Where is it produced? So you need to know all the 4 different types of hormones, and they give you some things that it could be. Now, looking at the question, we know that the glomerulus and the hypothalamus is not actually in the menstrual cycle. So you have a choice of 2, corpus luteum, and you have the pituitary gland. But of course, if you have revised, you would know that it's actually the corpus luteum, and I'll explain this in the next part. Describe the effect of high levels of the progesterone on the uterus lining during pregnancy. For 1 mark, you would have to say progesterone maintains the thickness of the uterus lining. So for the 6th-mark question, it's a really nice question because it talks about the menstrual cycle. Explain how the menstrual cycle is controlled by hormones and negative feedback. So there's so many things you can say, and the things in the mark scheme, they give you lots of different points you can talk about. But remember, it's quality of communication, full sentences, and punctuation, of course. Just in case you don't know anything about the menstrual cycle, and it's a 6-mark question, I'm going to explain it for you in detail so you know. So, the first thing I'm going to talk about is the pituitary gland, which there is 4 hormones you need to memorize off by heart. FSH, LH, estrogen, and progesterone, and where they are actually produced. So here it says ovaries and the corpus luteum. So the first thing is the FSH. FSH causes a follicle, which is this, and it causes the egg. It's basically around the egg, and it's basically there only to mature one of the eggs inside the ovaries. When this is released, it actually produces another hormone called estrogen. Now, estrogen causes the lining of the uterus to thicken and grow. When it actually grows and it's really thick, this actually then makes it stimulate LH, which is actually produced in the pituitary gland. So, FSH, we just said, this means FSH is inhibited. And then there's a rapid increase of LH. LH is made in day 14. This stimulates ovulation, and the follicle ruptures, and the remaining of the follicle develops into corpus luteum. This secretes progesterone. Progesterone maintains the lining of the uterus, inhibits LH and FSH, so you don't have multiple eggs being ovulated. Just in case I didn't make it clear, the follicle is actually the clusterous cell around the egg, and this ruptures, which then actually makes the corpus luteum, which then releases the progesterone. Just like the question we had before, and it's asking us about if a fertilized egg implants the uterus because a woman becomes pregnant, then the level of progesterone stay high. Now we need to explain why. The uterus lining has a thick, spongy layer of blood vessels. The blood supply allows the placenta to develop. Here's the placenta. And the placenta supplies oxygen, glucose, nutrients, and it removes things, urea, carbon dioxide. Now, levels of progesterone stay high just to maintain the lining of the uterus during pregnancy. The uterus lining has a thick, spongy layer like we just mentioned. So, we're on the last question, and it's about enzyme technology, which is another great topic to be assessed on. The diagram shows the human insulin gene can be genetically modified bacteria. So, it actually gives you all the stages, which is also wonderful. The human chromosome, and then it says restriction enzyme to cut the insulin gene from the human chromosome. Then it shows the insulin gene joint to the plasmid using an enzyme. And then it makes the recombinant DNA and the plasmid into the bacterium, and then it also has a different version here. The plasmid is removed from bacterium and cut with the restriction enzyme, and then you have the bacterium as well. So name the enzyme to join insulin to the plasmid. Now you need to know this as ligase. And the next part of the question says, suggest why the same restriction enzyme was used to cut the human chromosome and the plasmid. For the 2 marks, you would need to say 2 things. So the sticky ends, which leaves complementary bases, can pair up. This means it can hold the inserting gene into plasmid more successfully. So the last 6-mark question is also very beautiful. Enzyme technology can be used in the production of foods including sweets, vegetarian cheese, and lactose-free milk. So describe how the enzyme technology can be used to produce these products. So you need to talk about chymosin, sucrose, fructose, lots of different things. You have a lot of things you can talk about again. So let's start with sweets. Made by invertase, converts sucrose into glucose and fructose. This means it reduces the amount of sugar in the mixture, increases the simple sugar content, and it increases sweetness without having to put any more money. The production of the soft scented sweets, and there's a production of lower calorie sweets. And the next part is vegetarian cheese, as they ask in the question. It's made by chymosin, produced by genetically modified organisms. Chymosin is a clotting protein and reduces the rennet extract from animals, which is found in the gut of cows. Then you can talk about the lactose-free milk. So each single one here, you get 2 marks, 2 marks, and 2 marks. So it's made by using lactase, by immobilized enzymes. Lactose is converted to glucose and other forms. It's beneficial to people with lactose intolerance because they have a side effect from milk. For the last question, it's very nice as well. So washing powders can be used as biological and non-biological. So, describe how biological washing powders can make clothes cleaner than non-biological washing powders. So, you can say about digestive enzymes such as protease and pepsin, which break down protein stains from meat, that's incorrect, from meat, which are soluble. Now, this is converted into amino acids. So, we are making this video for Ask J Chemistry, our science teacher. And you can look in the playlist. We've made a C3 June 2014 paper as well, which you can go check out if you wish. And also remember to go to, there's also things you should know about electrolysis and titrations for C3 for Edexcel. But also make sure you go and check out Study Smart Science. And this has lots of different playlists as well, because you might check it out. And it's really good because it has AQA things as well. So make sure you watch the videos, write notes as you're going along, but also make sure you revise enough for the exam. And that has 155 subscribers, so go subscribe. Thank you.